Okay, so when we submit grants, we submit really sexy titles. So I'm sorry if anybody has turned up to see what serotypes we find post-intervention. We learned a lot in this proposal and in the work that we did, and we've improved our Salmonella isolation approaches and a lot of the techniques that we use in the lab. But I just want to say that up front so hopefully no one is too disappointed by the end of this. So... Okay, I'd like to give credit first to the team. This was I started at UGA in March of 2019, and USPOULTRY. This was the first competitive proposal that I was awarded as a new faculty, and it wouldn't have been without my colleagues who've since retired, Mark Barang and Nelson Cox at the USDA ARS, and an exceptional student, Surendra Razamseti, is a master's student, arrived in my lab in February 2020. From India, and I made him sit down for a month and read papers and said he needs to learn the foundation of what he does. And then a month later, COVID hit. So he accomplished all of this, including, I think, over 30 plant visits during a pandemic. So props to him. So we had two objectives to this work. The first was to improve Salmonella surveillance by addressing limitations in conventional culture detection. We published this a couple of years ago in Food Microbiology. And then the second objective, the second objective was to more accurate, sorry, I don't know why this is not coming up, to more accurately trace Salmonella populations at commercial poultry processing facilities. Pre and post intervention, and we published this last year in Poultry Science. So let's talk about the limitations. The most important limitation is how long it takes to isolate Salmonella. So there's several rapid methods out there right now for Salmonella detection, for Salmonella quantification, but for much of what we need to do, we need an isolate on a plate. That approach takes at least a week. So after the sample is received by the lab who's doing the culturing, if it's a plant isolate, it's going to be cultured for 24 hours in buffered peptone water. That's a non-selective broth that allows any injured Salmonella that have been injured during the antimicrobial use during processing to resuscitate, to recover. That's a step for 24 hours. Salmonella are then transferred into a selective enrichment broth. There are two broths that are typically used for Salmonella isolation, tetrathionate and Rappaport vacilliatus broth. I'll refer to that as tetrathionate and RV. That's a culture that's done for 24 hours. The purpose of that culture is to reduce the microflora and to allow Salmonella to grow. One thing that I've learned since I've been working on these selective enrichment broths is, as a naive postdoc, I always used to say these selective enrichments, they reduce the background flora and they allow Salmonella to grow. They allow Salmonella to hang on with a thread enough for us to select it. But Salmonella doesn't necessarily proliferate well in those. Then those samples are plated onto selective indicator plates. Colonies are picked, confirmed, and then characterized in whatever manner folks are using for characterization, whether it's whole genome sequencing or other molecular methods. The second limitation is that traditional culture, if you've heard me speak before, this is usually what I'm talking about, the traditional culture culminates in picking a single colony off of a plate. If you've got a really enthusiastic technician in the lab, maybe they're picking 2 colonies. We know that food animals often have multiple serovars of Salmonella present. So when you pick 1 or 2 colonies, you're picking the colonies that represent the most abundant serotype or member in that population. So in the example that I've shown here on the screen of the chicken, each of those different colored circles represents a different type of Salmonella, different Salmonella serotype. The gray ones are more abundant. So when you're picking a colony off of a plate, it would be that gray one that you identify. Let's go ahead and call that Kentucky. We know that Kentucky is prevalent in chickens and is not associated with human illness. And so when someone might eat a contaminated piece of chicken, that Kentucky might not cause illness, but one of those other serotypes that's in the background may. And so when public health laboratories are isolating those Salmonella, they find a completely different serotype than what we find when we're doing our routine surveillance of poultry products. And so it makes it really hard to track what's going on. It makes it really hard to solve the problem, right, in production. My lab developed a technique a few years ago we call CRISPR-Seroseq. It's a deep serotyping approach. It's a molecular method that allows us to identify multiple Salmonella serotypes that are present in a single sample. Our input for this assay is that overnight enrichment broth that I've shown at day 3 from the tetrathionate or the RV broth. That's a mixed enriched culture for Salmonella that would contain those multiple serotypes. So for this grant, to address these 2 limitations, our first goal, or our first potential solution was, could we take those day 2 and day 3 broth cultures and somehow combine those together? We kind of think about this as Goldilocks, so it's too hot, it's too cold, it's just right. Could we do anything to the pre-enrichment step to make it a little bit selective, where it wasn't too selective that injured cells wouldn't recover, but that they could be recovered, and we could shorten this whole protocol by 1 day. We call this selective pre-enrichment. The components that we added to our buffered peptone water were malachite green. This is a component of RV broth that reduces coliforms, and bile salts, which is a component of tetrathionate that causes oxidative stress in target bacteria. And we also use novabiocin. This is an antibiotic that's often used in BGS agar plates. And then on the deep serotyping side, the question we had was, we've always done this deep serotyping from that full selective enrichment. Would we be able to do that from the selective pre-enrichment? A paper that we published before we got this grant funded, we showed that we were not able to do CRISPR-Seroseq from a buffered peptone overnight culture. There's just too much other background flora in that that we can identify the major serotype, but we can't identify a full profile of serovars. For those of you who might be using the BAC system, so that pre-enrichment broth that Hygiena uses has a little bit of selection. We've had a little bit more luck with that, but that still is not selective enough to reduce that other microflora for us to profile the full collection of Salmonella serotypes, reproducibly, I should say. Okay, so this was our experimental setup. We take our sample, and I will say, well, our sample types in this project were slightly unconventional. Instead of using carcass rinses, so you're limited in how many carcass rinses you can do when you're doing a project. If you take 1 carcass rinse, is it really representing a flock of 30,000 birds? So one approach that had been used for Campylobacter by collaborators at USDA ARS was to use drip pan sampling, where you hold a drip pan under the line and you collect the drips while 500 or 1,000 birds cross that line. So there's pros and cons to both methods. That's the method that we used here. We still felt that we needed to do the recovery step for a short period of time to allow any Salmonella to recover if needed. We chose 4 hours because if this method was going to be implemented, we wanted it to be able to be completed within a technician's working day. So if the samples arrive at 10, 11 o'clock in the morning, that they could add selective components before they go home at night. So the first 4-hour incubation was in buffered peptone water. Then we separated that into the different selective pre-enrichment components. So we had buffered peptone water as the control. We had buffered peptone water plus bile salts and novabiocin, or malachite green and novabiocin, and then we added all 3 as well. And we incubated those for 20 hours. So we had the same 24-hour time frame for that pre-enrichment step. We then directly plated those from that selective pre-enrichment. And this was our test protocol for selective pre-enrichment. And we did this alongside the conventional protocol where we did the 24-hour culture in buffered peptone water and then went into TT and RV in parallel and plated onto XLT4. So this is the data. So these are from 9 different plants across the US. We also collected these samples post-pick. So we wanted to be able to see that when you had a high microflora, that we would be able to reduce that and detect Salmonella. So these were done at 9 different plants. We collected 2 samples in replicate from each plant. The top 2 rows on both tables represent the 24 hours in buffered peptone water, followed by the 24 hours in either RV or TT as indicated. The 3rd row in each table is the buffered peptone water alone, right? So that would be the least selective of this. And then the bottom 3 in each table represent our test selective pre-enrichment approaches. Salmonella is, any of you who work with it know that it's a beast. Sometimes it grows in RV, sometimes it grows in TT. Our experiments cost double because we have to use double the enrichments to make sure that we're finding it. So I've shown the data collectively. So collectively between RV and TT, 18 out of the samples, 18 out of 18 samples were positive. For buffered peptone water, 12 out of 18 were positive. And then for the combined selective pre-enrichment conditions, across all 3 conditions, we were also able to identify the 18 positives that we could find with the traditional selective enrichment approach. When we break that down by actual specific condition, you can see, so when you look at the gray columns on the left-hand side, there's greater recovery in tetrathionate than there is in RV, but we didn't get 100%. So I think it was 17 out of the 18 we were able to recover in tetrathionate than RV. Buffered peptone gave us a very low, only 12 isolates, only 12 samples were recovered. And then the black bars represent those different selective pre-enrichment conditions. So bile salts, so the first black column is the bile salts and novobiacin. Bile salts is a component of tetrathionate, and you can see that looks better than the malachite green novobiacin combination. But again, collectively, those 2 first black columns, the malachite green and the bile salts, give us 18 out of 18 positives. In some of the cases, and I haven't broken it down here, the 3rd option where we added everything in seemed to actually be a little bit too selective. So maybe that 4-hour recovery in buffered peptone water wasn't enough for those cells. We then did deep serotyping of these samples. So deep serotyping, like I said, is an approach to be able to identify multiple different serovars in a sample. We only analyzed those samples where we had a positive in either tetrathionate or RV and at least one of the pre-enrichment conditions, selective pre-enrichment conditions. We tried the BPW, like I said, there was too much background flora. On average, there were 2 serovars per sample. There are 44 samples here. The most we saw in any one sample was 4 serovars. Each of these colors on these bar graphs represents a different serotype, and the y-axis is the relative abundance of those serotypes within a population. So the yellow bars represents the most common serotype that we found, and that was Salmonella Kentucky, unsurprisingly. 7 out of the 8 paired samples, so when I say a paired sample, selective pre-enrichment compared to selective enrichment. So we wanted to make sure that the CRISPR-Seroseq data that we got from this new enrichment approach matched what we were getting with conventional approaches. So 7 out of the 8 paired samples had additional samples in the selective pre-enrichment that weren't found in the selective enrichment. And then we had 8 paired samples that had additional serovars in the selective enrichment, but not in the selective pre-enrichment. I see you smiling, Karen. You know, it happens. That's Salmonella. And then we had 4 out of 18 samples that were that matched. So overall, it is somewhat comparable. Typically, the major serovar that was found in one condition was found in its paired condition. Okay, to summarize this part, so selective pre-enrichment is able to successfully recover Salmonella from all samples where we were able to isolate Salmonella from a selectively enriched sample. And so it shortens the protocol by 24 hours. Deep serotyping shows that overall the serovar populations across those different enrichment approaches are comparable. And if I had to pick one, I would pick bile salts and novobiocin. We got better recovery with that than we did with malachite green and novobiocin, but we don't recover everything. So we're still, we need to use RV and TT to find everything. It's not surprising that maybe we need to use two different selective pre-enrichment approaches in parallel as well. I'll move on to the second objective. You can see it now. So the goal here was to use this new approach, selective pre-enrichment, to then track Salmonella serovars through processing. So again, we did this with the dip sampling, and to sample at multiple points during processing. That's dip sampling, I mean carcass drip collections. So we visited, or my student visited 3 different plants for this study. He visited each plant 6 times. And at each visit, he took samples from post-pick, post-IOBW, post-chill, and then he collected pre-dip parts and post-dip parts. And he did this in duplicate on each visit. So 18 visits and then done in duplicate. You can see across these 3 plants, we're seeing, as expected, right, a reduction in Salmonella presence after intervention. So at post-chill, we see a large reduction compared to the post-IOBW. We see a little increase in pre-dip parts, which others have seen as well. And we see that reduced in the post-dip parts as well. This is using the conventional technique. When we compare the selective pre-enrichment to the selective enrichment, we can see that those outcomes are somewhat similar, except for a couple of situations. So it was interesting to us that in the post-chill samples, so these are samples, obviously it was very low incidence, right? So this is not a statistically meaningful observation, but I think it's a pretty cool observation. For the post-chill samples, we were only able to recover those Salmonella using selective pre-enrichment. So if you think about it, selective pre-enrichment is not as harsh as potentially using RV and TT full on. And so maybe those cells were injured, they'd just come out of the chiller, right? And so we were able to recover them with that selective pre-enrichment step, but not with the full enrichment step. Conversely, when you look at so those are indicated in the red boxes, when you look at the largely pre-intervention samples, so that's post-pick and post-IOBW, there seems to be slightly better recovery when you're using the selective enrichment. And that also makes sense because you've got a lot of background flora that are present there. Maybe you need that full-on RV and tetrathionate to be able to knock down those microflora so that you can detect the Salmonella. Again, not statistically meaningful, but I think it's an interesting observation. We did do CRISPR-Seroseq on this. I'm not going to show the data because the idea was to track how things moved through processing. We got very few positives in post-chill. We just did CRISPR-Seroseq on plant number 3 because that was the only plant where we got positives at each of the 5 locations that we were testing. But as you all know, anybody who's done these experiments, you'll get a positive that was in post-chill, but you don't have the negative that came at the stage right after that, right? Or you get a positive in your post-parts dip, but you didn't have a pre-positive. And so it was just very difficult. I think we had 2 positive post-chills where we actually had samples upstream that we could sample. And so it didn't give us anything meaningful in terms of selection of serotypes through this process. But what we did do was to look at the samples where we had paired serotype data for the selective enrichment, the conventional approach, and the selective pre-enrichment, and we compared those populations to each other statistically. So for this, we used the Bray-Curtis method. This is a method that accounts for both the presence and absence. So did I find the same serotype in both media conditions, and it also accounts for the relative frequency of those members. So if I found Serovar A in condition 1, was it 90% of the population, but if I found it in condition 2, was it only 10% of the population? And so this statistical metric can make those comparisons. A strong match is anything that I've highlighted in dark yellow, and a moderate match is anything shown in pale yellow. We only had one pair where there was a sort of complete disconnect between the two media types. And so the take home from this is that these culture approaches, when it comes to deep serotyping, are recovering the same serovars at the same relative abundance. So in summary, Salmonella incidence decreases as we go through the different intervention steps in processing. It's higher in pre-dip parts than in post-chill carcasses. Deep serotyping shows that the serovar profiles between the selective pre-enrichment and selective enrichment samples taken at those 5 different stages during processing are comparable. And similar serovar complexity, I didn't show this, but similar serovar complexity between post-BIC and post-IOBW, but reduced complexity between IOBW and post-chill. I will say that the 2 most common serotypes that we found were Salmonella serotype Kentucky and Infantis. Those were the most common, but I didn't show the data for the caveats that I shared with earlier. So with that, I'll finish and say I have first and foremost, thank you very much to USPOULTRY for funding us. This was, like I said, my first competitive grant I got when I got to UGA, gosh. Thanks very much to Surendra. He's now a vet working in North Carolina. He did all of this hard work, and again, within the confines of working in a pandemic and doing all of these plant visits, did an incredible job. So, and then to Mark and Nelson, who've been wonderful collaborators. I'll take any questions.